My name is David Bean. I'm a graduate student in the lab of Dr. Manish Sagar, and today I'll be discussing how a prior SARS-CoV-2 infection, more than a COVID-19 vaccination, associates with a lower incidence of a subsequent symptomatic endemic coronavirus infection. It's now been well established that a prior SARS-CoV-2 infection or a COVID-19 vaccination boosts the immune response towards SARS-CoV-2 so that it decreases the risk of reinfection with SARS-CoV-2, at least for a few months, but also decreases the severity of COVID-19 disease. What's not currently well understood, though, is whether these immune responses towards SARS-CoV-2, do they cross-react and can they prevent against future infections by other related but different human coronaviruses, such as ones that may circulate currently in the human population or novel ones that may emerge in the future. And so our objective of this study is to examine the incidence of and immune differences against these related but different human coronaviruses among individuals with a previous SARS-CoV-2 infection, a previous COVID-19 vaccination, or individuals without either type of SARS-CoV-2 exposure. And so in order to answer this question, we can look further into our methods section to see how we recruited patients from Boston Medical Center for this study. We divided our cohort based off of 3 separate clinical histories. 1, if the individuals had a previous SARS-CoV-2 infection but no history of a COVID-19 vaccine. 2, if they've had a previous COVID-19 vaccine but no history of a SARS-CoV-2 infection. Or 3, if they've had neither SARS-CoV-2 infection or vaccination. We identified these individuals because between the months of December 2020 and October 2021, they came to BMC with signs of a respiratory infection. At BMC, they often undergo this respiratory pathogen test to identify which pathogen is causing these respiratory infections. And so we're interested in 2 potential outcomes from this test. 1, if they test positive for an endemic human coronavirus, such as 229E, HKU1, NL63, or OC43. Alternatively, if they test positive for a different respiratory pathogen, for example, such as rhinovirus. And so we're able to analyze these test results and look at the incidence of respiratory infections dependent on the time since SARS-CoV-2 exposure, which I'll discuss in Figure 1. At the same time, we're also interested in the immune responses elicited by either a SARS-CoV-2 infection or a COVID-19 vaccination and whether they cross-react to the endemic coronaviruses. And so between the months of December 2019 and October 2021, we collected blood samples from a subset of individuals within this cohort prior to or without a symptomatic respiratory infection. And so with these blood samples, we are able to test a wide range of immune responses within these individuals. And so in Figure 2, specifically, we're going to look at patient plasma and its ability to neutralize pseudovirus particles that express coronavirus spike proteins. And then in Figure 3, we're going to look at levels of T cells within these individuals that are able to recognize and become activated in the presence of various coronavirus peptides. So first, we want to look at the incidence of respiratory infections post-SARS-CoV-2 exposure. In Figure 1, we'll first focus on the incidence of a non-coronavirus infection. And so on the left, we have a survival plot, where on the x-axis, we have the number of days between the SARS-CoV-2 exposure and to when the individual came into the hospital and tested positive for a respiratory infection. On the y-axis, we have the percent of individuals without a non-coronavirus infection. And so how the survival plot works is over time, as more and more individuals come into the hospital and test positive for in this case, a non-coronavirus infection, this percentage goes down. And so we wanted to compare the incidence rate within each group over time, and so we used a cost proportional hazards model. We have adjusted this model to account for various comorbidities that may be associated within each group to account for increased risk associated with a respiratory infection. And so ultimately, when we compare the group of individuals with a prior SARS-CoV-2 infection to those with no prior experience, we see no difference in the risk towards getting a non-coronavirus infection. And we see a similar trend when we compare those with a prior COVID-19 vaccine to those with no prior SARS-CoV-2 experience, both represented by a hazards ratio of around 1. We see a slightly different pattern, though, when we look at the incidence of a endemic coronavirus infection. So now when we look in green at those individuals with a prior SARS-CoV-2 natural infection compared to those in blue with no prior SARS-CoV-2 experience, we see a hazards ratio of 0.48. This lower value represents a decreased risk of getting an endemic coronavirus infection is associated with having a previous SARS-CoV-2 natural infection. Though when we look at having a previous SARS-CoV-2 vaccination, we don't see really any difference between that and those individuals without any prior SARS-CoV-2 experience. These clinical results suggest that having a previous SARS-CoV-2 natural infection helps protect against a subsequent endemic coronavirus infection, whereas having a COVID-19 vaccine does not elicit the same response. And so next we wanted to look at the immune responses to see if we can start to figure out what's mediating this protection. In Figure 2, we're focusing on a component of the humoral immune response, specifically the ability of patient plasma to neutralize or prevent the infection mediated by a coronavirus spike protein. So in this assay, we're mixing patient plasma with pseudoviruses that express a coronavirus spike protein to see if they prevent the infection of a target cell. Here we're calculating area under the curve, which is a measure of neutralization over a series of dilutions of patient plasma. A value of 1 represents a strong ability to neutralize the pseudovirus of interest, where a value of 0 represents an inability to neutralize the pseudovirus being tested. In panel A, we're testing pseudoviruses that express a SARS-CoV-2 spike protein. What we observe is those individuals with a previous SARS-CoV-2 natural infection or with a previous COVID-19 vaccination have a better ability to neutralize these SARS-CoV-2 pseudoviruses than those with no prior SARS-CoV-2 experience. Next, when we tested pseudoviruses that express endemic coronavirus proteins, for example OC43 spike or 2G9E spike, what we observed is there was really no difference in the ability to neutralize these pseudoviruses between any of our three groups. These results suggest that neutralization alone may not be the driving factor that is mediating the protection against a subsequent endemic coronavirus infection caused by having a natural SARS-CoV-2 previous infection. Now in Figure 3, we're focusing on the cellular side of things, looking at CD4 positive T cell activation after stimulation with peptide pools derived from coronavirus proteins. Here in this assay, we're mixing cells derived from patient blood with peptide pools that correspond to a specific coronavirus protein and measuring T cell activation after 24 hours of stimulation. And so within this assay, we're looking at levels of T cell activation in relation to our negative control or background levels of T cell activation. So a value of 1 represents no difference between our experimental condition and our negative control, where higher values represent greater T cell activation in relation to these coronavirus proteins. And so in panel A, when we stimulate our cells with peptides derived from SARS-CoV-2 spike, we see increases in CD4 positive T cell activation only in those individuals with a previous SARS-CoV-2 natural infection or a previous COVID-19 vaccination. When we stimulate cells with SARS-CoV-2 nucleocapsid peptides, we only see increase in T cell activation in those individuals with a previous SARS-CoV-2 natural infection. Next, when we stimulated these cells with peptides derived from OC43 spike now, we see increases in T cell activation in both groups that had a previous SARS-CoV-2 infection or a COVID-19 vaccination. Interestingly, in our last panel D, when we stimulate cells with OC43 nucleocapsid-derived peptides, we see a slight increase in T cell activation only in the group with a previous SARS-CoV-2 natural infection. In conclusion, we showed that a prior SARS-CoV-2 infection, but not a COVID-19 vaccination, helps protect against subsequent symptomatic infections by endemic coronaviruses. We showed that this protection is likely mediated by immune responses to proteins outside of the coronavirus spike protein region, highlighted by our results that showed increased T cell activation towards coronavirus nucleocapsid proteins, only observed in those individuals with a natural infection with SARS-CoV-2. These results highlight that the addition of coronavirus non-spike antigens may be used to help improve the efficacy of future pan-coronavirus vaccines. And so with that, I would like to thank all my funding sources, but more importantly, I would like to thank all the patients of BMC for donating blood to this study. And so with that, if you have any questions, feel free to email me at djbean@bu.edu.